All right, ladies and gentlemen, welcome back. We are looking at Part 5, proteins and nucleic acids. Just two more of our biomolecules that we're going to be looking at in a little more depth. These are the functions. Proteins are really important in our body because it pretty much carries out every single process that we have. We have the little acronym, which is HEATS, which should help you remember the 5 different functions: hormones, enzymes, antibodies, transport, structural. So just as a quick thing, so hormones are kind of messenger signals, and we'll be looking at these in more detail as we go on. Enzymes are going to help speed up reactions, and we'll be having a whole video on these next. Antibodies help to target infections, so foreign cells for destruction. Transport, things like hemoglobin is a transport protein, and what this does is these ones carry oxygen. And then structural ones are pretty important, things like keratin. We have things like collagen, which is in your skin to give it some structure. You know those people that get those collagen implants and look like they have big slugs on their lips? Yeah, that's collagen. Might have spelled that wrong. Actin and myosin, which are both used in muscle contraction, and then also plasma proteins that we're going to talk a lot in when we get into the cell, all the different proteins that are involved in allowing what enters and exits the cells. Okay, so just as always, if you need to stop it to take a quick peek and write a few things down, feel free. What is a protein? All it is a polymer of amino acids. So a polymer of amino acids turns into a polypeptide. So this is just a long string of amino acids, and then once it folds and forms bonds, then it's going to be an active protein. Okay. So here's an example. As you can see, this is one amino acid. One amino acid is going to bond to another one, and then it forms this long chain. This is something that I'm going to hand out to you guys in class. You guys will have a amino acid handout for all the different types. Amino acids, we're not going to go into a whole lot of detail yet on these, but all you need to know is that there's 3 parts. There's an amino group, which means NH2. There's an acidic group, which is a C double bond O OH. And then there's also a remainder group, which we call the R group. And all amino acids are going to be the exact same with regard to the first 2. They're all going to have an NH2. They're all going to have a variation of the COOH, but all their R groups is what's going to be the big difference here. So as you can see, here's an R group for arginine. It looks dramatically different than one for histidine. Okay, but if you look, it still has its NH groups, and it still has its COOH groups. Okay, moving along. What are polypeptides? Just as a quick little recap, it's a single chain of amino acids. What joins these amino acids together are called peptide bonds. Okay, and we went over this in organic chemistry, but if we want to make peptides by joining these amino acids, water is going to be leaving in each situation. So it's a dehydration reaction. If we want to go the other way and break it down into single amino acids from these polypeptides, that's going to be a hydrolysis reaction. Proteins can be organized into 4 different organizational levels. We have our primary level, so this is not a protein yet. All it is a linear polypeptide. It would have no function right now. Secondly, the secondary level organization is when it starts to coil, and it starts to coil into these things called alpha helix or a beta sheet. Okay, here's the little abbreviation for alpha helix, and for beta it's just a B with a longer straight line. Most won't still have a functionality either after the secondary. It's not usually until it gets to a tertiary where the twisting finishes and all the bonding finishes as well. So this is where not only will you have peptide bonds, but you can also have ionic, covalent, hydrogen, and some disulfide bonds. Okay, and then the last one, this is more rare. It's a quaternary bond, or a quaternary level of organization. This is where you actually have full functional proteins that link together. So something like hemoglobin actually has 4 polypeptides that are all linked together. Okay, so this is kind of like if you guys ever watch Transformers, where you have like the single cars or the guy can transform, but then when they come together, they form this like superhuman robot. It looks pretty much nothing like that, but you know, you get the gist. Okay, moving on to nucleic acids. Nucleic acids, we have 2 types. You guys have probably heard this before in Bio 11. You have DNA and RNA. DNA stands for deoxyribonucleic acid. RNA is ribonucleic acid. Compare and contrast, DNA is double-stranded versus single-stranded RNA. What does DNA do? The whole point of this is it stores all the genetic information. In other words, all the codes for the order of amino acids is due to DNA. So it's going to tell you what order amino acids go into. Why is that important? Because that dictates what the polypeptide does, and that dictates what protein is going to be created. So different set of DNA, different protein. How is that different than RNA? RNA, all it is it's a messenger. It takes that DNA code and we change it into a new language called RNA, which is better, more easily able to transport, and it goes to the ribosome where amino acids are placed in a specific order based on what that DNA told that RNA, and that's going to tell you what your protein is going to be again. Okay. And we'll be seeing this a lot more when we get into protein synthesis next, or in 2 units from now. Okay, so what is a nucleic acid actually composed of? It's composed of polymers of nucleotides, just like proteins were made up of polymers of amino acids. And this is a nucleotide, right? This group here mixing together with something else. Okay, we'll look at it in more detail in a second, like right now. So here it is. So we have a phosphate group, which is PO4. We have a pentose sugar, which is a 5-sided sugar with 5 carbohydrates, and then also a nitrogen base. Okay, these nitrogen bases are going to alternate, and they are complementary, which means that they only pair with certain things. So if you look at this, it's going to go from phosphate and sugar are going to be connected, and it makes up like if you're looking at like a ladder, it would make up the ladder rung. Okay, and then what joins these 2 sides together are our nucleotide bases, and we're going to go into some more detail on this in class as well. So this would be your G's going with C's and your A's going with T's. Okay. Difference between RNA and DNA: RNA does not use thiamine, the nitrogen base; it uses uracil instead. It's an evolutionary process where this RNA is just a little bit more basic than the DNA. Okay, so they're very similar in structure, but they have different functions. Lastly, finishing off, ATP is our energy source. We call it adenosine triphosphate. Adenosine just means it's a kind of a hybrid between the adenine nitrogen base and the ribose sugar, and triphosphate means 3 phosphates. Like I said, it's our energy molecule, whereas if you're breaking down 1 glucose, it's going to produce 36 ATP. And the reason why we don't just use glucose is because it's too much energy. You don't need 36 ATP to carry out certain functions. And here's the chemical reaction. In order to do that, ATP gets broken down into adenosine. This is diphosphate and a single phosphate. And when that bond is broken, it releases this energy. Okay, so that's it. Maybe let's see it on the outside here. Just make sure you pretty much just know the structure and functions of the 2 biomolecules, proteins and nucleic acids. Make sure you know what the monomer is that makes it up. So for example, amino acids and nucleotides. And make sure you just generally know some of the details that we went over. So if you have questions, which I'm sure you will, we'll bring them up next day. Thank you.